Hello, everyone. Welcome on in. Today we are live with Dr. Marah Wabeh. She is a former early career leadership program scientist, a member of the GSA, and now is going on to bigger and fancier things. So, Marah, please, would you introduce yourself and let us know, where you are right now, who you are, what makes you tick?
Yeah, that's a big question. The last one, maybe we'll slow the roll there. But yeah, as Flynn says, my name is Mara. I am a scientist by training, so I have a PhD in genetics. I recently graduated, so I've only been a doctor for 6-ish months. I did my research on genetics of schizophrenia while I was in grad school, and right now I have transitioned over the last 6 months into science policy. So I manage government affairs and science policy for the American Society of Pharmacology and Experimental Therapeutics, or ASPET for short. Um, yeah, and it's been a wild ride, but loving it so far.
So, that is already like an amazing career journey that you've laid out for us of getting like what your PhD was in and all the way to like what you're working on now. So, Marah, take us back to the beginning, right? How did you know that you wanted to work in something like schizophrenia? Probably you didn't know early on, but when did you think, what set you on that journey? Where did your love of science start?
Yeah, that's a good question. I feel like if I had to trace it back to before grad school and before any of that of what got me into science, I feel like even as a child, I've always been just a curious person and an observer by nature, just inherently. I liked observing people, I liked observing things, trees, animals, you name it. Just by, again, inherently, that's just kind of my personality. Um, then my both of my parents were scientifically inclined. So my dad is a pediatrician and my mom is a math teacher. So um, there was always talk about science and philosophy and whatnot in our household. So that was something I was kind of brought up in. And then you go to school, and early on in elementary or middle school, people teach you about the scientific method and how to employ that. And there was that, again, curiosity of like, oh, there's a way in which one can take on observations and things they see in the world and analyze them more and learn about them more in a way that has been laid out and studied. That's another thing. I am someone who likes rules and likes following directions and easy ways to make things happen. And science felt like a way I could, again, employ my curiosity and observation in a way that felt structured for my brain, and it made sense for who I was and how I functioned. And I think in high school was the first time I was ever introduced to genetics as a concept of heredity and Punnett squares and all that stuff. And it was very cool. It was very interesting. And like I said before, I was someone who was interested in people. People fascinated me in their behavior. And then I went to college and just started learning a little bit more about genetics and the ways in which there are connections between behavior and biology and genetics. And that was something just in the back of my mind as I went through um, undergrad. I didn't do any behavioral or schizophrenia genetics related stuff. I did join my undergrad lab. I did fruit fly work, which was pretty cool. I loved my mentor and I loved the scientific community, the scientific research community at the time. It was my first time ever doing anything like it. And I loved it. So I started looking into exploring getting a PhD and doing more research in the future. And that's when that idea in the back of my mind of combining human behavior and biology and genetics in one place. And I found my way to schizophrenia.
Nice.
That was the journey.
So, in your undergrad, that's really cool that you're able to be in a research lab. Do you remember how you ended up joining a lab and what was the research, like as an undergrad, right? I feel like when I was looking at for a lab, there was just too, it's overwhelming. There's so many options. Like how did you end up picking the topic, and what was it, and what did you study in undergrad?
Yeah, so it was a coincidence actually how I ended up in my undergrad lab. There was an open house, like a research open house in the biology department of, I think it was like middle of my sophomore year in undergrad, and it was at a time when I think I was starting to think about where I could take my degree, and I wasn't sure. I knew that I didn't want to enter like professional school. We're talking like med or dental school or anything like that. But I didn't know enough about the opportunities within biology that where I could take my degree. And I stumbled upon this research open house where graduate students in the Department of Biology at my undergrad institution were just presenting their posters and their work. And I just walked around and was attracted to one particular poster. The grad student of the lab that I ended up working with at the time, the work of the lab. So I guess I'll try to describe it in simple terms. So humans, we males and females. Males have one X chromosome, and females have two X chromosomes. And the way in which nature kind of balances the difference between us in order to still have us be the same is one of the female's X chromosomes gets suppressed genetically, so it's a lot of the genes are not expressed. What's cool about fruit flies is the males also are XY and females are XX. Instead of suppressing one of the female's X chromosomes, the males actually double their X chromosome expression. And that was the work of the lab. And I was just very fascinated by the idea of like, there's this similarity between us and fruit flies in terms of the number of chromosomes, but in the way mechanistically we function to balance that out is similar in ways, but different in others. And it was very intriguing. So I just approached the grad student. I was like, is there a way I could get involved? And she connected me with her PI and Dr. Vicki Miller, and she was an incredible mentor to me. She kind of guided me through, she trusted me with a mini project of my own that, looking back now that I've done a PhD, it was a very small project and nothing crazy, but at the time I had never touched a pipette ever in my life and was learning how to do that and measuring things and being around fruit flies and all this stuff. It was very cool. I was very intrigued. And again, the scientific community at my institution and in my lab was just very welcoming and very open to me asking questions. And I never felt stupid asking questions, even though I didn't know much at the time. And I feel like I learned a lot about science and about the scientific method and about applying sort of hypotheses and experimentation and things like that. And yeah, after that experience, I was like, I want to continue to do more of that. And even though that changed slightly during my time in grad school, again, which led to my interest in policy and where I am right now, but that was kind of where I was at during my undergrad and what led me to apply to grad school.
Nice. It sounds like a really good environment to learn in, where you can, ask the questions and not have to fear that there's any kind of judgment associated with it, right? That's a very unique kind of place.
That's what I always say. I think mentorship and good colleagueship and environments are truly what inspire us to either stay where we are or want to do other things. It's the people around us that kind of push you in one way or another. Absolutely.
Yeah, it can definitely drive you in both directions, right?
Sometimes negatively, but
yeah. Do you remember what your project was? Like anything about it? You said, like, looking back on it, it was a small project, but I'm sure for some of the people out there, even this would be like a pretty big and cool project. Like, you're talking.
I remember I did a lot of PCR at the time. There was particular. So I'm on the X chromosome of the male fly. There are particular genes that are expressed that have been identified to associate with a specific complex that help with that double expression. And there were satellite regions also within that X chromosome that have been found to be associated mechanistically with the process. And there were questions about read through transcription between two specific regions, and it was just me doing a bunch of PCR, trying to see if they do link or not. So again, like looking back, it may have not been as big of a project, but for me, not know having done PCR ever, it was really cool.
I mean, that's amazing. You're looking at X chromosome dosages in different
Yeah.
sexed animals and trying to figure out why it's opposite from us and how that affects the. I think that's really cool.
Yeah. Again, like the contribution to the general project and just how much I learned being in that environment scientifically and just overall was fascinating. It was very, very cool.
That's awesome. And so you went from that, and then now you're applying for graduate school, right? So take us through that process. Like, how have you already had to identify a lab at an institute? Did you have rotations? Like, how did you end up in the lab that you did your PhD in?
Yeah, so that process was a lot at the time. So I knew I wanted to do genetics because I had experience in my undergrad doing genetics, and had talked to people within my department just asking, like, where are genetics programs? Where do people think I should apply? And from recommendations from people and just doing my own research on Google on like where are genetics departments in the US, I went through maybe a list of 15 or so, and just remember vaguely looking at their websites and like looking at what professors did in these departments. I remember specifically like if there it was a very heavily like computational or a very heavily ecology sort of programs, I strayed away because I knew those avenues were not exactly I wouldn't fit in with that personally. I tried to find programs that, or PIs within those programs that had more like of a molecular biology lens to genetics. And I think I picked like 8 programs total and just applied and hoped for the best. Once the process of the application happens, many of these schools invite you to come interview for them. So you get an opportunity to go meet with some of the people that you may have listed in the application of like, oh, I'm interested in their work, and be in the city where that school might be to see if it could potentially be a good fit. So I did a lot of interviews, and it was actually, looking back, it was a fun time. It was a really cool opportunity to just like be flown around and meet people from all of these places, from different programs that offered different things, and have people just talk to you about their research and meet fellow sort of prospective PhD students who had very similar interests and were looking to get into similar programs. So it was a cool time. And then I was accepted into multiple programs and just had to sit down and do a pros and cons list of the location, the research of the people that I talked to. And then one very important thing, and I think that was one of the main, I would say, drivers for where I ended up going, was just like how happy people seemed in the program. Because prior to going to grad school, people told me, and I still, even during that time, didn't grasp what it was until I was in it, of how difficult the graduate school journey is, and being around people or peers who seemed a little bit happier was important to me, because I'm like, okay, if they seemed a little bit happier, that means the program is comfortable and there's more hope for me to also be happy in the program. So yeah, those were some of the criteria. And then the first year in graduate school, I think most programs do it in this way, where you basically rotate into different labs. So my program specifically, our rotations were 3 months long. So we spent a lot of time in the labs that we did. So I chose 3 labs with very different work and in very different places, even physically on the Hopkins campus. That's where I went, and just sort of got a feel for my relationship with the mentor. Do we get along? Is our communication style the same? Do I like the work that I'm doing? Do I enjoy the environment of the lab? Do I like talking to my peers? All of this stuff, and that's how I ended up in my genetics of schizophrenia lab.
Do you remember the other like? So what were the organisms like? What were the basic topics that you were doing in those
Yes, so
rotations?
all of the work that I did was in human cells. So that was something I also was strategic about. I wanted to do translational genetics work, so I chose labs that were pretty translational in their work. So I did um, all of my work in tissue culture, and my schizophrenia genetics work, my lab, we did it in induced pluripotent stem cells. So throughout my rotations, I worked in cell culture, and for my PhD as well, I kind of remained in that.
Cool. So it's just you go from the flies to the cell culture.
Yes, it was very different. And I think, again, like during that time, it was intentional. Like I loved working with flies, but I don't know if you've ever worked with flies before. It can be a lot maintaining, feeding them, and and all that stuff. Even though I didn't realize at the time that doing tissue culture is equally as much maintenance. Stem cells, I had to feed them every day. So the maintenance work didn't become less with translational work, even though that was part of what I thought what I was getting myself into.
And then actually we have a tying kind of question is would it have been
Yeah,
possible to do the two things together? So like can you express a gene in other model organisms to mimic schizophrenia?
that's a good question. So, yes and no. There are mouse models for certain schizophrenia, I would say, that replicates schizophrenic behaviors. So they can mimic anxiety like behaviors, they can mimic certain things, but do we have anything that 100% replicates what schizophrenia captures? No. I think part of it also, which is important to note, we still don't know 100% what capitulates the genetics of schizophrenia in humans. Like we are still, as a field, parsing through all of the variants and all of the genes that are involved in increasing someone's risk of schizophrenia. That's also another thing. Schizophrenia as a disorder, like many neuropsychiatric disorders, is very complex, and it's not genetic only. So there's environmental factors and genetic factors, and an interaction between genes and environment that results in someone sort of having it. So can we 100% recapitulate any of it? No, both because we don't fully understand it, and two, because animals, however close they are to us, we can't fully recapitulate behaviors in them the same way that we see in
So
humans.
what was the I guess then the benefit of working with the cells? Because like with the difficulty of cells, right, is that you can't tell you anything. So like I'm sure there's
Yes.
difficult. Well, neither can a mouse, right? But so like what kind of drew you towards the cells in terms of translation? Like what about those cells make it easier maybe to work with this kind of question?
So that was one of the things that I liked a lot about the work of my lab. So one of the things about schizophrenia, again, it's a very complex disorder, and there are many, many, many variants, over 200 variants that have been identified thus far to be associated with increased risk. And these variants are common, or what we call common in nature. So chances are me, you, many people we know have some of those variants, but obviously I don't have schizophrenia, neither do you. There is a specific combination that, again, we still are, as a field, trying to parse through what it is. Individually, we still don't fully know what each individual variant can do. So this is where the work of cells, if you can isolate a specific variant and try to study it in a very simple cellular environment where you're not distracted by, where it is not distracted by the organism as a whole. Because sometimes, if this variant is causing something cellularly in an organismal sort of environment, things can balance itself out, and you don't really know what it does. So when you isolate that variant in a pure cellular environment where there is no balance with other cells or interaction with other things, you can capture what that one change can do genetically and molecularly and mechanistically in order for us to study the overall interconnection between those variants, because we don't know fully what they do. I hope that made sense.
So I feel like that could be difficult as well, right? It could be a double-edged
Yes.
sword. So like you isolate the variant, and you
Mm-hmm.
to see like what kind of schizophrenia you have. It might be these combination of variants, right? And so you look at this individual one in a dish, but that dish might be is that like a monoculture of cells? So is it how
So
close is it going to be to the actual human brain, I guess, right?
it's, again, it's not in the sense of like, it's not in the sense of like it'll recapitulate behavior, but it'll recapitulate what this change does molecularly. So if we could understand what it does molecularly on a very, very simple, small level, then maybe we could grow out from that into a bigger connection of like, if this variant does this and this variant does that, how do these two talk to each other in order to create more molecules or more proteins, and then we could grow out into behavior, if that
So
answers your question.
yeah, so is that something you can do in the dish as well? You can do multiple variants in one dish and see then like after you've isolated them how they interact.
Absolutely. It's, again, it's very difficult. Like, doing one variant alone took me 6 years of the PhD. So that's also another thing. Like, I think technologically we are still advancing, and we are getting better in the way that we manipulate genes in cells, in dishes and stuff like that. So hopefully in the future there will be spaces for us to sort of do multiple variants at the same time. I think it is doable. It's just definitely a lot of work, and would take more than one grad student to do it.
Well, so yeah, tell us about your work. Like how did you get hooked on that project? What was the day to day like? Because you're telling us like this one variant took so long. Like, what are the difficulties? What are the easy parts? Like, how did you get hooked on this question? And yeah, just tell us about it. Tell us.
Yeah, yeah. So again, like, the question was from, that was part of the conversation with me and my advisor when I met my advisor, of the type of work that he was looking to do. And I was very interested because, again, it was a way for me to explore that element of like human behavior, and into in a very specific biological way. The overall work was very cool, but as I've said before, one of the I don't know if I said it before. One of the challenges was technically that it was very specific and very niche, and in order for the work to kind of, it needed a lot of time to grow. And I made sense of that as I was in the PhD more and more. So I loved doing the work. Like, one of the things I loved the most was being in tissue culture, and because it was work that we did every day. We would feed the cells, we would lift them up and look at them, and all this stuff. It was very relaxing. Like, I meditated a lot during my time in tissue culture. So I liked the work. The day-to-day, like I said, was fun, but over time it did make me realize that the impact of my work is going to take years, and maybe not within my lifetime that I would be able to fully see what it does. And you could say that was one of the things that drove me to think about other. Oh, sorry about that. To think about my career and how I could take all the things that I was learning and loving about being a scientist and working in a lab, and still contributing to that environment in a way that felt less niche and less specific.
And also
Yeah.
maybe feels more immediate, almost.
The impact more broad and more direct than I felt during my PhD. Yeah.
We did have a question in the chat if an
Yeah.
advisor is equivalent to the PI of how you're talking about that.
Yes. So a PI is a principal investigator, which is a person who essentially manages the grants for the lab, and they are also my advisor. So the principal investigator on the grant and the project is also an advisor, and they are synonymous.
Okay, so you mentioned that you were working on stem cells, um,
Yes.
and these are I believe you said the pluripotent inducible stem cells. Can you tell it
Yes.
what does that mean for those at home? Like, these aren't like from embryos, right? These are a different kind of stem cell.
No.
How does that work?
Yeah, so they're essentially cells that are skin cells, other blood cells, other types of cells that are taken from donors who give some of samples of their cells, and they are taken into the lab and basically given specific instructions by, we give them proteins or genes, and are fed specific nutrients, and they take those instructions internally and they sort of revert back in status. So, a skin cell is a very specific cell that expresses certain things, and it's a very specialized cell. But when you suppress certain expression of genes and take away certain proteins, it's no longer as specialized, and it takes away some of the specialization. It looks less specific. It's vague in in what it has, and it reverts to that pluripotent state, which is essentially a state of a cell that you could take that cell and turn it into something else. So when you take that skin cell, you make it into an induced pluripotent stem cell, and then you could take that induced pluripotent stem cell and redirect it, give it instructions again to have it make any type of cell you want it to. So for us, because we were studying schizophrenia, so the brain, neurons were an important cell, so we would take those induced pluripotent stem cells, give them the correct instructions, feed them the correct things, and they would re-specify to becoming neurons.
So you could take any patient's cells, right? That you could sequence this patient's genome. You knew exactly where the mutations were. Take their skin
Yes.
cells and make neurons out of them to try to model what's going on in their brain.
Yes.
That's amazing. That's so cool.
It is, again, it's super cool. Like conceptually it's very cool. Doing it on the daily was very cool. But like I said before, it is very specific. And as a field, like we have grown so much in the last 10 years, but there is still a lot of work to be done in order for us to get in any place where we feel like we fully understand or capture the disease. And again, that is what drove me to want to do science policy.
Yeah, you mentioned that you were working on that one variant. Could you
Yes.
elaborate on what that meant? Was it just from one patient you were trying to figure out what was going on? And I guess what does that look like? Like what did is you just say, big congrats again on defending your thesis.
Thank you. Thank you.
What did that culminate in? Like how did you know when you've identified what that variant does? Like what does that end result look like?
So I actually had two projects. One, I worked on a variant, and well, I worked on two variants. One is published, and people can read it. That one came specifically from a family. So we identified a family with history of psychiatric illness, and they it looked like they had a autosomal dominant inheritance, which is not common because schizophrenia is a complex disorder, and it that type of inheritance is very specific to Mendelian disorders or disorders where you have one gene that is involved in the disease. Schizophrenia has many variants that are involved. So it was a curious case per se. Within schizophrenia, there are some variants, maybe like a handful or under 10 or so. I can't remember the exact number off the top of my head, that are large deletions or duplications. And those have a very high chances of increasing risk of someone developing schizophrenia. So overall, schizophrenia has over 200 single nucleotide variants that are associated with increased risk. But that increased risk is very small. Again, they're common. I probably have some of them. You probably do too. They don't increase it by much. But then there are a few that increase risk by so much that, again, their inheritance can look closer to Mendelian disorders. And that is what we were curious about. We were wondering if this family had a specific mutation that resembled those large duplications that we see. And we did actually identify one variant in a specific gene that has been associated with neural work and stuff like that. So what I did is I if people are familiar with CRISPR-Cas9, which is the genome editing tool that scientists use. So I used that tool. So we have stem cells that they're just they're just like healthy people stem cells. So somebody donated, an unknown person to us, donated stem cells. So we have these pluripotent stem cells where the mutation doesn't exist, basically. And I used CRISPR-Cas9, the gene editing tool, to introduce that mutation into the gene that we are interested in studying. And essentially had cells that had no mutation and cells that did have a mutation. And then we did some RNA sequencing and calcium imaging and other stuff, and compared the non-edited cells with some edited cells to see what changes molecularly does this variant do. And the overall conclusion was it looks like it's doing something that could potentially be a causal, but I guess we could never fully 100% be like, yes, this is the thing that is causing it, because it is complicated, but there is evidence that indicates that it is involved, and sort of like encouraging the field overall to explore more of what this gene has to offer, potentially for schizophrenia risk.
So we have a question from the chat of why does schizophrenia most often express itself in individuals late 20s, or other psychiatric disorders like bipolar express themselves around puberty? Did this affect your research at all?
That's a good question. I actually don't know the answer to that. I do know that with schizophrenia, there is sort of difference between when males and females have onset. Like males tend to have onset of schizophrenia in their late teens, and females tend to have it in their early 20s. I can tell you that with bipolar, like there is a lot of overlap genetically with schizophrenia. Questions of why it has earlier onset, I honestly don't know. It can affect research if you're doing like research on participants or things like that, but in terms of cellular work or molecular work, it doesn't make as much of a difference. If you're doing computational work, so if you're doing large genome association studies, it also doesn't matter as much because you only need like cohorts of age-matched controls and people with a disorder. But I don't know the answer to that question, but now I'm curious too.
I had another question for you. Given you're saying that you and I might have some of these precursors, right, or variants that could contribute to the schizophrenia, but we don't manifest as clinically diagnosed. How could you ever know your healthy cells that you're comparing to were fully healthy, right? Like if you were to insert that mutation, is it possible it pushed you over the line, right, and got you a stronger phenotype than without? Because who knows what's in that healthy individual?
I think that's a great question, and there are ways we could sort of proxy that. We call it a polygenic risk score. So it is a score that is sort of created from combining all of the variants that are known to increase risk or to contribute to risk, and it's weighed by how much they contribute to risk, and every cell is given a score after you. So a low polygenic risk score is like you have less of these variants, and a high polygenic risk score is you have more of these variants. But again, because it's still very early of our understanding of how many more of these variants there are and what they do and what they don't do, I wouldn't say like we could use this score to fully diagnose like, oh, this person with a low polygenic risk score definitely is fully healthy versus someone. It's a little complicated, but there are ways to proxy what that could look like. So cells with lower polygenic risk scores probably have a lot less of these variants than a cell with or a line with many more of these polygenic risk scores. It is more likely to resemble schizophrenia genetics than a low polygenic risk score cell.
That's really cool.
I hope that answers the question.
No, that's definitely, that's really cool that you can account for all these like variance is built, and it makes sense that it's going to take time to further know because we don't know all the variants and how they might interact.
Yeah. Yeah.
What was your favorite experiment or result?
Ooh, my favorite experiment by far was my calcium imaging experiment. So it was the only really visual experiment I got to do in grad school. Most of my work was doing PCR and RNA sequencing. So visually, I didn't deal with like pretty images very much. But towards the end of my journey, I did some calcium imaging, which is essentially a way to measure how much the cells are talking to each other. So neurons, as we all know, um, essentially like carry electrical signals, and that's how they communicate with one another. And the more active neurons are, the more of these electrical signals they put out, and the less active they are, the less of these electrical signals they are. And as scientists, there are experiments that allow us to measure that activity and visualize that activity. So with the calcium imaging, I get to take my neurons in a dish and put them under a camera, a live camera, after I've treated them with some stuff that allows me to visualize their signal, and then watch it happen. Watch the signals glow green in between, and stop and not stop. And it was very beautiful. It took a long time, and it was very it took a while to get the experiment to work, but it was incredibly rewarding, and I loved it. Was one of my favorite things, like watching the neurons flash green and take the images and take the recordings. Yeah, so I would say that was probably my favorite experiment, despite being one of the hardest experiments I did in grad school.
What was the difference between a schizophrenic cell line versus a healthy one in terms of the flashing? Did they flash more?
So it wasn't, again, I wouldn't call it the schizophrenic cell line because it's it's tricky. I would call it the one with the variant. They're, the edited cells versus the unedited cells. So the edited neurons actually had more activity at a certain point than the non-edited neurons. It wasn't significant enough throughout, so I did the measurements over a period of time. I think they started about the same, and then the edited cells spiked up midway through quicker than the unedited cells, but then they both plateaued later. So it seemed like there was the pace at which they were maybe potentially growing. We still don't fully know. There's more questions to ask there. That made that difference, but we did see a difference. If that answers your question.
Cool. We do have a question in the chat as well. It's how do you determine that if someone is schizophrenic for your studies? Do you rely on neurologists or psychiatrists for the original diagnosis?
Psychiatrists. So all of the family members that we, again, have learned about the family, it came from a group of psychiatrists. So multiple diagnoses from different psychiatrists who see these patients and interact with these patients. And by the time it comes to the research lab or the principal investigator, it had already been sort of talked about with other people. Yeah.
Do you do that for your control healthy individuals? Do they also have to go through psychiatric evaluation, or is it just someone?
So that's actually a good question. I don't know, because a lot of the cells that we get come from cell banks, essentially, that we just order from NIMH or other cell banks that are available. I'm not sure if the people who are donors are diagnosed before, but they just like tell you if this person is healthy or has a disorder. Sometimes they have other things, like they don't have schizophrenia, they could have other disorders, but it's still considered healthy for our purposes, because for what we're looking for, any other diagnoses probably don't matter as much unless it's psychiatry related. So if they had anxiety or something else, we would probably be interested in knowing. But I don't know what the process is like for getting people diagnosed in these large banks where people get cells.
Yeah, I imagine it gets more and more complicated as we figure out more things as well, and more variables you want to test for. And so if you never tested to begin with before it was banked, you'd have no idea.
No. Yeah.
It's a really hard field that you were in, and it just kind of illustrates like all that we've talked about. Like it wasn't straightforward experiment. There's a lot of things that could go wrong. There's a lot of variables you have to account for, and it kind of encompasses as to like how Herculean an effort of one variant of these E's. Like we see like how many different things you have to test, and
Yeah.
it's really really cool that you're able to
Thank
find
you.
out though what this variant did.
No, it is. And again, like there's still more work to do. Like what I did is still, I would argue, is step one into exploring what this gene and what this variant can offer the schizophrenia genetics field overall. Yeah.
What would you say are the next steps then that should be that if it were up to you to direct this research, what would the next steps be?
Yeah, I think so. My What I would be curious about, because when I did the experiment, we introduced multiple variants into that gene and explored multiple. I would probably want to like hone in on one of those and see which one is actually the one causing the effect that we saw. I'd also be curious the interaction with other proteins, because there's a whole complex that the gene, so our gene makes a protein that interacts with other proteins in a complex, and in that complex there are other proteins that their genes have been associated with schizophrenia. So I'd be curious one if one is like is already a known schizophrenia gene, the one that we've identified as potentially a good candidate for a schizophrenia gene. I'd be curious what else is going on in that complex, and if other genes in that complex could be potentially relevant for psychiatric disease overall. Because I know the cascade of that mechanism, there are other psychiatric disorders involved with some of those proteins. So the whole complex is of interest, if that makes sense.
Nice, like going from the individual gene level to like building a gene web essentially and
Yes.
figuring out
Yes.
how they
What happened?
interact.
Yeah.
Yeah, nice. And so, Marah, you
Yeah.
mentioned, you had a different interest that grew in graduate school, and
Yes.
that was in science policy. Could you tell us a little bit about um, where that interest came from and what exactly, like how that came up, what exactly that meant to you, and how that, I guess, changed the course of a career?
Yeah. So my journey to science policy was not, I would say, like an easy direct line one. I feel like I discovered science policy through a bunch of things that happened to me during grad school and landed me exactly, I feel like, where I meant to be. Um, Essentially, my first year of grad school, similar to many, I would argue, graduate students feel, there were a lot of imposter syndrome feelings that grew during that first year. I was one of the very, like, I think there were few Arabs in the School of Medicine overall at Hopkins, but not in my immediate environment. None of my cohort mates and none of the people that I was interacting with were Arab. So I didn't know many people who were like me or had a similar background to me. And that led to even like larger feelings of isolation during that first year. So
Do you feel like that's a general problem in the sciences and not just specific to an institute?
for sure, for
And
sure. I think there's an element of like being in graduate school alone is isolating enough, because I think which I don't think we maybe talk about enough, the PhD experience is very unique and very specific. And if other people in your family or people around you in life outside of graduate school have not experienced it, it can already, that alone can make you feel very isolated. And then combine that with this sense of like an identity crisis of some sort, of like feeling like there are very few of me in this space. It amounted to strong feelings of isolation during that first year. So I started essentially just like seeking out opportunities to help change that, whether that is student wellness or DEIA work. And I got involved very heavily my second year in student government at Hopkins in the School of Medicine. So I was on the Graduate Student Association. I was on their diversity and inclusion committee. I did some work with that felt very inspiring to me. So I've always been someone who liked working with people and had more brainstorming sessions and sitting together and just discussing things. And I did a lot of that during my time in the Graduate Student Association. I felt a lot of fulfillment in the work that I was doing, engaging with others. And that's when things also started to click for me that in the lab, even though I loved the work that I was doing, I was spending a lot of time on my own working in the lab. My lab mates were great, but a lot of our work needed that like zero in focus on what you were doing. So I was spending a lot of time on my own in the lab doing very specific niche work that I knew at the time like I would not maybe be able to see the impact of my work on the broader society. But then on the side, I was also working in the Graduate Student Association, having all of these talks and engaging with people all the time and seeing some of my work be implemented right away by engaging with university admin and seeing again some of the impact of the work that I was doing like directly. And that's when the shift started happening a little bit of like, okay, what can I maybe do that involves teamwork and leadership? And I had no idea what science policy was at the time. So it started all of like this feeling of like, here are some things I like to do. I like to talk to people. I like to be in meetings, and which is wild because some people could argue like that's the last thing they want to do in life. But I liked it. I liked being in meetings. I liked being in control of a project, which was also part of my graduate school experience. I liked having my own project. I liked leading a project. I like management, having steps and instructions to follow. So there were just these vague things that I was like, I like doing all these things, but like, what does that mean? I don't know. So I went and talked to the career office. I literally went to the career office and was like, I don't know what I want to do with my PhD, but here are things I like to do. And it was as vague as I just described it to you. It was simply like, I like to be in meetings and I like to talk to people. And that was it. That's all I gave her. And she was like, okay, well, how about you just come to some of the panels that we have on careers and careers outside of academia and things like that. So I was like, okay, great. I'll start going to those. So I started going to panels. Hopkins had hosted many of those where they'd bring people from different paths and different career opportunities and just share their journey, what their day-to-day looked like. And I would keep note when people would talk about their day-to-day, when it's like, oh, I have a lot of meetings. It's like, okay, I like to do that too. I do a lot of brainstorming sessions. I do a lot of writing. I do a lot of reading. Like people would describe things and I would take note of whichever one of these careers and people are talking about matched some of the things that I already knew I liked to do. And policy kept coming up a lot. So I started exploring, okay, like let me see what this science policy is about. And at first it was incredibly intimidating. It was very broad. There are a million things that fall under science policy. But I realized that a lot of the work that I was doing with student government is policy. Now, is it particularly science policy? Maybe not exactly, but it was, higher ed policy. I was engaging in policy work on a very small scale. So I was like, okay, how can I explore that broader, explore what policy means within science? And that's when I started seeking out the GSA work. So scientific societies, that's just a tip for everybody, are a great place to start when you want to explore something. Like go to your society and see what they have to offer you. And ECLP program and the Science Policy Committee was one that I wanted to kind of explore. And I learned a lot. I loved working with GSA and I loved working with Jessica. We did some science communication work also at the time, but it all still fell into that larger idea of like, here are things I like to do, but I still don't know what it means to employ them practically into what science policy is. because I would read and I would talk to people, and everyone had different answers for what science policy is. There's federal policy, and you could work for the government, or you could work for a private industry, and it just felt so incredibly broad. So I went from, not knowing at all what I wanted to do, but having things that I like to do, to identifying some sort of career path that seemed to match up the things that I like to do, but not knowing much about it, to doing ECLP and feeling like, okay, I have a better grasp of what policy and science policy is. I took the course with GSA as well, with FASEB at the time, learned more about what that meant. And then it was like, okay, how can I tangibly like touch that and see what that looks like for me day to day? I did an internship for 3 months at a policy consulting company.
Was that
So I
during
took a
was
leave
that
of
during
absence.
graduate school?
Yes, I took a leave of absence for 3 months in grad school and did a full time internship um, with a consulting company. And it was a little bit of a culture shock, but I got to experience what it was like to be in the workforce, even on a small scale, as a policy person, as a contributor to the science policy field. Even though it wasn't everything that science policy was or everything that science policy offered, I got to be in it and engage with people in a way that was very different from science and what I had been used to. And it just solidified my love for science policy and the work that I thought was meant for me. Um, and yeah, it grew from there. I was like, okay, yes, I definitely want to do science policy. And I started applying for jobs after grad school after I finished. And here I am.
So what was the experience like? Like what was this internship like? Like what was your day to day when what because we bring back the point of like what is science policy? So what did that
Yeah.
definition end up being for you that made it like you telling us the little things like the meetings and the project management and the interaction with
Yeah.
people, but what became the definition of policy that really drove you finally towards that?
Yeah, yeah. So essentially, like how I would define science policy, it's that bridge between scientists and policymakers. You have people on the Hill every day, the Congress members who make decisions for how much funding agencies get to have every year, what the distribution looks like. And that's kind of like the start, the higher up from there. You have staffers who, some of them have science backgrounds, that recommend things to Congress members. Then it trickles down to the work of the agencies. So NIH, FDA, there are scientists in those institutions that also work to advocate for more science funding or for specific issues that policymakers sort of control. And then it can trickle down even more. So some of the work that we did in my consulting experience, where we would get contracted by federal agencies to do internal policy related work for them. So it wasn't federal policy, it was policy for the agency. But because it was a science related agency, it still fell under the larger umbrella of science policy. So this is why, again, it's really complicated, even as someone who's in it now, to describe what science policy is, because it is incredibly broad, which is why I take it back to even like the work in my, at Hopkins in my grad institution when I was doing student government work. That was policy. And then it grows from there, from like your institution. How does your institution scientists talk to agencies? That's science policy. How do your agencies, the work of the agencies, talk to federal congressional members? That's science policies. Where does your society, so our scientific societies where I work right now, how do they connect scientists who are members that they advocate for to federal and congressional employees on how to bridge that science policy? So it's making sure that things trickle all the way up and all the way down from those who make decisions about, whether it is how much funding scientists get to have, to how many agencies there are, and bills about drug policy or how animals in research are sort of controlled. they make the decisions, but how it trickles down all the way to the simple policy of like internal work policy, that's still in how I see it, all under the umbrella of science policy. So depending on like if you're somebody who's interested in the broad idea of science policy and you're just unsure of where your niche fits in, that's something I'm still trying to figure out, even as someone who's in the field of how much impact do you want to have? How broad do you want your impact to be? Do you care to be a federal employee or not? Like, these are questions people can ask themselves as they're exploring that, but policy is all the way. Even as a graduate student involved in your own institution, policy, that still functions, in my opinion, as science policy, because you are engaging as a scientist with policy work, even on the higher ed small scale, to contribute to the scientists in your community of other fellow graduate students. I hope that answers your question.
Yeah, no, that's I've never heard it explained that way before, and I've never thought about it that way before, but it makes total sense. Where in the web were you doing your work for your internship?
Yeah, so we were, again, so you have policymakers, you have agencies. We were contracted by federal agencies like NIH and NAGRI and all those institutes to investigate DIA work for them. So internal work on the agency to improve some of their sort of work. So they would contract the company that I was working for, and we would bring in the expertise to be like, here is a research project for like 3 months. We could look into what you could do better internally, and then you provide recommendations, and then they take it, implement it internally to make their institute better, so that again it's better for the people in it, and then the people in it could continue to do the work that they need to do for the where it goes higher and lower, and it trickles back and forth. So that's kind of where it was. I would say it was like on the agency below level.
Cool. So we have a question from Chad that I think kind of dovetails into this. So when, because you mentioned you took a 3 month leave to be able to do the internship, were you ever
Yes.
worried that working in policy during grad school could have had an effect on the future career as like a scientist in like a postdoc sense by some of the old guard that is in academic industry? And how did you, I guess, maybe overcome that fear, and how did you deal with it?
So I think it's a two. I would say it's like a two answer question for me. One, I am very lucky and grateful that my mentor was someone who was very supportive of whatever career explorations I wanted. So I never felt any pushback from my own advisor, and that provided a safe environment for me to even bring up the fact that I was like, oh, I want to take a 3 month leave out of the lab, which I know is not the case for many, because not all advisors are that open to letting their student just leave the lab for 3 months. So I was lucky in that way. But I think the other part of it, in terms of like how it would have affected my career broadly, I think with policy specifically, there is a shift in the way scientists are thinking and are talking about policy, in that it is no longer this like separate entity of science and policy, and they're two different things, which is a good thing. I think that recognition of, science is political, and that engagement from scientists to policymakers and vice versa is crucial and very important, also helps. So if I was in an environment that was generally, shifting in thinking of like policy and science are interconnected and we need to be involved. And I was very lucky to have a supportive advisor who didn't make me feel any type of way. I think the other thing is I also knew I was not going to stay in academia. So there was also the element of like, if there are people who thought some type of way about it, I didn't care because I was leaving the academic space. Like, I wanted to contribute to scientists and science overall from a policy lens. And by the time I made the decision to do the internship, I knew that. I knew I wasn't going to look back. The question was like what kind of science policy I wanted to do at that point. So it was easy to not look back in that way. But again, I was lucky to have had a very supportive advisor and have done it in a time when I think a shift in thinking, both overall and like what scientists can do with their degrees, and also with that science policy interconnection.
Yeah, so the definitely back to the good mentoring that it seems like you've had along the way really tied into the success currently. So,
Yes, absolutely.
Marah, what skills did you need to be able to apply for the internship? So if someone out there is really wanting to be taking part in something like this, how did you go about even how does one become competitive for it? Like what does one need to do to find something like that?
Yeah, I think so. A few things. The first thing I did was talk to people. So the all the people who came to the panels that I went to during my first couple of years, I would just connect with them on LinkedIn and tell them what I liked about their talks, and reached out to them and was like, hey, can I just have a conversation with you on your career path and what that looked like? And having I had so many Zoom calls with people. It was also during 2020 when I had nothing else to do because our lab was shut down for like 4 months. Um, and I would ask them, like, what makes a competitive intern to you? If I were to do a science policy fellowship, what do you think would make me competitive? And the answers that they were giving me, so things like get involved as a, just as a citizen. So go to your scientific society. If they do hill days, if they get involved with going to having virtual or in-person meeting with any congressional staff, get involved, ask them to be a part of it, because as a graduate student you are a scientist and your voice matters. So you could display and showcase that as a citizen of whatever um, district or state or city you are, that you are engaged in policy from a science lens and from a science perspective. All the student government work that I did, so demonstrating that I can lead a meeting, I have the leadership skills to do it, doing the ECLP work, so taking the science policy course with ECLP, doing the multilingual seminar series with Jessica, the process that took, how do you build a network, how do you engage with others, all of those skills from leadership to project management, which some of those you already do in the lab, just demonstrating how it could translate to a role that is not lab related. It all matters, because in a science project, in the PhD or anywhere within the science environment, you are responsible for a project, you have deadlines, you can manage yourself in a deadline, you've never missed a deadline. All these things are part of the work in policy that are all translatable skills. So from the student government work to the ECLP work, to just being engaged as a citizen with policy work. And again, the easiest way to do that, if you're unsure, is your scientific society. Most scientific societies have advocacy work that you could get engaged in some way, shape, or form. Writing, reading, demonstrating that you've written stuff, whether that is scientific or in a blog post somewhere. So you are someone who has the capacity to engage an audience in writing. If you've ever been to a conference, you are someone who's able to engage with an audience verbally. All of those things that one may not think are relevant, if you are able to kind of strip them from their context and be like, I can speak, I can write, I can manage a project, I can lead a project, all of those things are translatable and make you competitive for a policy internship or a fellowship. But I think again, like, you just got to demonstrate your interest in policy in some way.
Nice. And then did you feel that the internship was critical to getting the full-time position that you have now?
Yes. Yeah. Even, it may not have been, I don't know, because I did do the internship, but I didn't do a fellowship. So there's that. Some people after grad school will apply to like year-long, 2-year-long fellowships like AAAS or ASHG or elsewhere. And those tend to be where a lot of people make the transition. But you don't have to have a fellowship. I didn't get one, but I think the specific experience that I got in my 3-month internship played a role in me, I feel like, knowing more about what it was to be in science policy. So in my interviews, I didn't feel like I had this vague understanding of what the job was going to be. I knew what the job was going to be. So it made me a stronger interviewee during the interview process, because I was not only determined to get the job, but also knew what I was talking about because I had applied it. So I think it's less so that it made me competitive in terms of what it looked like on my resume, but I think it made me more confident, and a better communicator for what I wanted to do because I had already done it. It gave me the experience to feel more confident to do it.
With the PhD, do you feel like you need that degree to get involved in science policy, or can someone get involved with a master's degree or a bachelor's, or like what is the level that you need to get involved?
People can certainly get involved at every level. I think the PhD is helpful because there is an element to showing you've had the expertise as a scientist. So, being in a PhD, we're talking spending 6+ or 5+ years in working on a very specific project, learning how to critically think and dissect information and apply it and implement it. It gives you that expertise. So if you have a PhD, it is seen as a way to showcase that you have the experience and you have the expertise. But I don't think only PhDs are or should get involved in science policy. You could definitely get involved at every level. It's just a question of how much your involvement will be based on the expertise that you have and what you can demonstrate. Yeah.
Cool.
There are many who don't have PhDs in science policy.
Okay, well, I mean, that's reassuring that there is more ways to get involved.
Absolutely.
What is your day to day like? What does a person in science policy do? Like,
Yeah, so we do a lot of advocacy work. So my day to day looks like a lot of meetings, as I mentioned earlier, something I like to do. So we do a combination of things. I'll speak for what I do in a scientific society as someone who is on government and science policy staff. We work, there's like member interfacing work, and then there's advocacy and Congress interfacing work, or agency interfacing work. It all involves around the general sense of like advocacy. So you have members that are all scientists that have work in specific areas, that are interested in specific areas. So many use animals in their research. Some do drug research. All of those things are controlled by Congress and how it's controlled, and who gets to have access to these things, and the funding for all the agencies comes from that. So we sort of support our members, or support the scientists that do all that stuff, by advocating for them in Congress. So we will have Hill days where we would go to the Hill and talk to congressional staff about the importance of science funding, the importance of animals in research, advocate for specific issues that our members bring to us. So with that, like, it looks like actually physically going to the Hill and scheduling meetings and being with other members. It also, my day-to-day looks like doing a lot of educational work. So we have a program where we bring in early career scientists to teach them about science policy. So again, expanding the network of scientists who are engaged and want to do science policy work. So explaining what Congress is, and talking about federal agencies and all the issues that are of interest to scientists that fall under our society. There's also work in engaging with federal agencies. So places like NIH will sort of ask the public a lot of questions about things that they do. So changes in their policy, changes in things that how the agency runs its work. They call them requests for informations, or RFI. So they post these to the public, and they're like, we are creating this change. What do you all think of it? So as a scientific society who are members, some of these changes will impact the work of the scientists and that we support. We will engage with those requests. We'll take surveys from people in our membership, and we have a science policy committee with expertise in those areas that could help in engaging with that. So we kind of try to bridge the gap between policymakers and where the policies are being made and the scientists who end up doing the work that policy impacts. So whatever policies impact our scientists and whatever science could be used to impact policy, that talk is filtered through some of the work that we do.
Wow, that seems like you get really, it's another web almost built on top of the web that you were saying earlier.
Yes, absolutely. Again, there are a lot of moving parts, and it's very exciting. Yeah.
We have a question from the chat. Are there science policy members that work for big pharma or other of these, I guess, industries?
Absolutely. There are sort of regulatory work that can be done. I don't know enough about it because I was never like super interested in it to kind of look into it, but absolutely. Because again, like even within the big pharma, you have people who have to monitor what policy changes could impact the work that they do, because they are large research labs in big pharma. They have to abide by regulations and abide by rules that are set for them from the federal government. And so, yeah, definitely they are around. And
do you have to interface with other potential science policy advocates as well? And how does some kind of interface like that work? Does it make things more complicated for you?
No, we definitely engage with other science advocates, people in other societies that do similar work that I do in my society. It's all a large network because they also are interested in some of the same stuff that we do. It's just maybe some of the issues are different based on the research that their members do, but the overall message and the overall goal of getting more scientists engaged in advocating for the things that we need in order to get our work done, starting with funding that is the general one that everybody is on board with, advocating for science to have funding, but other issues collectively as science policy advocates or science advocates overall. I think there is a lot of crosstalk, and I think that was one of the things that was attractive to me about the science policy world. The networks are very interconnected, and everybody kind of knows everybody, which is really, really cool. It made me feel less isolated than in my scientific sort of academic world, where I had to, again, work on a very specific niche thing where there's very few people in the world that maybe are working on the same gene or on the same protein. There are many people doing advocacy and science policy work that we engage with and interface with all the time.
And that engaging and interfacing is much more like a collaborative effort versus being competitive, right? Because that's different maybe from academics, where if people in your same field, it could be you're pushing towards the same like publication, so you might not talk a lot versus, like how... If you're talking with someone from a different agency, I guess this is just a different world for some of us. Like, what are the incentives for talking with them? Like, what is the benefit of working together? Is that encouraged? And how does something like a very simple question, but how does something like that happen?
Yeah, it's definitely encouraged because again, we all have the same goal. So like, if your organization has tips on how to run a good Hill day, we'd love to. You could share it with us so we could increase our engagement, and vice versa. If we both have an issue that we are both interested in, and we could, put our efforts together. Because again, like with larger numbers and larger voices, and the impact, that's how impact happens. If I'm one person going to the Hill telling congressional staff that we need funding for science, like I'm one person, I'm not going to make an impact. But if there's more of us that we could bring each other together and have the same message and talk to one another, then our impact is a lot bigger. So absolutely, yeah.
That's a really cool and nice to hear that that's kind of the interface happening.
Yeah.
Because I mean,
I'm only speaking from like the nonprofit scientific society world. I don't know how things get if I were to work for the federal
Hmm.
government or if I was working in like an industry. But and again, I would guess that it is all sort of still within that very collaborative environment, at least from some of the people I've spoken to who work in those. But I can at least speak from that environment that collaboration is definitely encouraged and present.
So because you're working with a scientific society, does that mean you're not a federal employee?
No, I am not a federal employee.
So if we had a shutdown, for example, that doesn't stop your
I would
work.
not be affected.
Because
No,
that's
a lot
something,
of
right, with
members would be, but yeah.
because like we hear that with like it's science policy, and so I think the built-in assumption is that you're on the hill, like you were saying on Hill Day, and that almost has that connotation of if
Yeah,
there's a
yeah.
shutdown, you also have to go home, right?
Yeah, no, but and I think that was what if I have like one message to give on this stream is science policy is so broad and it does not mean that you have to be on the hill or a federal employee to engage in it. You could still contribute very much to science from a policy lens in many, 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 many ways and in ways that may be more comfortable to you than others. So absolutely.
That's and actually because I pulled that up because you mentioned that when you were deciding of how to pick where
Yeah.
in science policy to go, you even mentioned like federal employee status. I'm like, huh, that sounds like I want to ask about that because that sounds really
Yeah.
interesting.
Yeah, yeah. Like in terms of like why I didn't want to be or
Yeah, yeah. Like what made you choose a policy versus federal versus maybe something
yeah.
else in that
Yeah,
team?
I think a couple, at least for now, because that could change in the future. I'm still sort of, I'm still new to the field. But in it at the time when I made the decision, part of it was, um, the pace of the work from what I've spoken to people, because government work is very complicated and there's a lot of layers and hierarchy and all these things. Things can be a little bit slow from what I've been told, and I'm someone who likes to be busy. So, especially coming from the academic world where we have no time to rest, I was working 24 hours all the time and going to lab at 7 p.m. So, I didn't know what rest was. So, the idea of like working in a much slower environment was a little bit intimidating for me. And I felt like I'm not sure that's where I want to start. Like, maybe I should start a little bit easier with something more balanced. So that was part of it. I think another part of it is because government work is impacted by policy on the federal level. So changes in, Congress or changes in the presidency or things like that can impact a lot of what happens all the time internally and projects and acts and things like that at least at the time, because that again could change, felt like I don't know if I want to enter that space of not knowing exactly how things are going to be for, especially with we have elections coming up next year, and I just graduated. I'm like, I don't know how things are going to look like, and I don't know if I want to get involved in that just yet. Maybe in the future, but things do change based on policy and vice versa with science and vice versa. So I was conscious of that as well. Um, but yeah, that was kind of it. And then again, I've done a lot of work with scientific societies. Again, I've been a member of GSA, and I've done a lot of work with GSA my last few years. And I felt like in some way I wanted to give back to the scientific society world because it had given a lot to me, and it felt like a good place to start to kind of again bridge the gap between still being a scientist and engaging with scientists and also doing policy work that was politics adjacent is what I like to call it.
it's it's really wise of how you picked the field you ended up in, like, and you're just going through like all these different extra steps, and like
Yeah.
you're saying, the regimes that are in charge, right,
Yeah.
and are going to affect those jobs, and then why you go and pick the society over federal, and it's a really cool and shows like how much you really can, like the field as an options of how many options you have, but also how much you have to think about and variables to account for when choosing which avenues you're going to pursue. So that's,
Absolutely.
yeah, we really
Absolutely.
appreciate that answer because that
Yeah.
shows again breadth, but also then you have to go to the depth level on that.
Yeah, yeah. And again, just kind of for anyone who's interested, encouraging people to know that, and which is something I had a hard time with in graduate school, because our whole lives, or my whole life at that point, was very structured, and like I knew what the next step was going to look like. So imagining this whole field where there's so much and there is really no right answer for where you can end up. It's just kind of like you got to trust your gut in the moment and kind of seize opportunities as they come. That's the best way to do it, because that was another thing. Even with my internship, initially it wasn't exactly the internship that I wanted. So there I was applying at the time, and there was another internship at a scientific society that I really 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 wanted, but the timeline of that internship didn't work. And the initial offer from the internship that I ended up doing looked different. I was going to work for a different department, and I was like, I don't think I'm fully aligned with the work there, and maybe I shouldn't do an internship. And the career office person at the time encouraged me. She was like, just tell them what you're interested in and see what happens. Like, advocate for yourself, tell them you have very strong interest in science policy, and see what happens. And they were so nice. They switched my whole internship. That I was going to work for a different department. They switched me to the policy and programming department. So, and I got exactly what I wanted. So that's another thing. Advocate for yourself. Be flexible. Like, there is space to learn things in places you may not have known were going to be for you. Like, that internship was the best decision I made, even though at the beginning I was very hesitant about it. And yeah, it's just like seizing opportunities as they come. There's no perfect answer, and you may not know exactly what title you're going to have or where you're going to work, but start by like just identifying things that make you happy. And it could be as vague and as simple, like I said with me, of like, oh, I like talking to people and leading a project. Lean into that, and it will lead you where you're meant to be. That's, I feel like, how my journey was.
Nice. I really like that kind of wrap up sentiment to it.
Yeah.
What is the favorite part of your job, and like the favorite thing that you do?
Yeah, would it be I don't know, like a loop if I was like, oh, I like bringing more scientists into policy.
No, not at all.
That's definitely it. I love engaging more scientists into policy. I think we don't have enough scientists engaged into policy. We have so much expertise in the science world, and so much to offer scientifically and otherwise. And I wish more of us engaged in policy in ways that I think would not only impact just scientists and the scientific enterprise, but like our world as a whole, because we do so much in our niche little world that are meant to impact the world in positive ways. And I think we could use a lot of that energy to engage broadly more with policy, to make the work that we do have a little bit more of a global impact. But yeah, absolutely, get more scientists into policy, get more early career scientists to want to switch careers to policy.
I mean, I guess in that vein, do you have any advice for those out there who want to get involved? Like, what at each career stage, like, if I was an undergrad student, grad, postdoc, what can I do? And
Yeah.
even if I want to continue in academics, are
Yeah.
there ways that I can get involved anyway?
Absolutely, absolutely. Even as an academic, or if you are thinking about it, you're like, I want to engage and I'm not sure where to start. As I said, like, starting with your state, every single state has congressional members and elected officials that would love to hear from you and want to hear from you as a scientist. If you don't know how to do that on your own, look up scientific societies. They have the resources to help take you to the Hill, whether it's virtually or in person, will help train you on, talking points and things like that, to help you engage with that process and make you realize that it's not as intimidating as you think it was. I mean, that is how I started with GSA. The first time I ever did a Hill Day was with you all. And it just felt like, oh, wow, this is a lot less intimidating than I thought. You just have these meetings and you talk about your research and why it's important and why the government should continue funding the work that you do because it matters to you and it'll matter to the world in ways one, two, three. So starting there, but also I think doing work on a much smaller scale in your own institutions. I think if there are issues within your school or within the space, the community work, like in your community, things that you feel passionate about, engage in it. Your engagement in policy in any way matters and it can translate to that work. So again, if you feel some type of way within your institution about DIA, get involved with student organizing and try and help shift the narrative and change the ways in which the scientific culture engages with itself and engages with the people in it, because that also contributes to bringing in more people into the science world, and more scientists mean maybe more scientists that engage into policy, and getting policymakers on board with funding the work that we do and engaging with it in positive ways, and maybe one day getting the public to trust scientists even more if we engage with them the correct way, and all that stuff. So I think there are many ways from the small scale work all the way to going to Hill Days and engaging with congressional work. But just, I guess, ask within your community, ask within your scientific societies, and there's always ways to do that. Yeah.
And is there any way for non-scientists to also get involved that you would suggest?
In science policy,
Yeah.
also absolutely yes. So if you are someone who has a family member who has cancer, or has been somebody who is impacted by specific disorders that scientists do study. So there's a research community for every disease and every disorder that we probably know about. So if you are someone affected by any of those things, or know people who are affected by any of those things, there are communities around them that also engage with scientists through, again, I'm thinking about Hill Days because it feels like one of the things that bring in the patient community and the science community and the policymakers and all of those people together. But yes, even if you're not a scientist, but your life is impacted by science all the time, whether it is through a disorder or whatever it may be. I mean, we all experienced it collectively in 2020 with the COVID-19 pandemic. We are all impacted by science and the work of scientists. So there are definitely ways for anyone to engage with science and scientific work and policy, even if they're not scientists themselves.
That's awesome. Thank you, Marah.
Yeah.
All right, y'all. And if anyone has any questions, feel free to drop them in the chat. I think we're gonna go ahead and start wrapping up, because Marah, we don't want to take up too much of your time. We thank you so very much for being here today and for answering our questions and taking us through the career journey. Is there anything?
I think this was cool.
No, I think it was awesome. Is there any last hitting message that you'd like to let people know about either science policy or your career path, and things that maybe, the little tinkle of wisdom, as it were, that you'd like to tell people?
That's a good question. I think the biggest thing is, like, it's okay not to know. If you are in an undergrad or a grad student, or somebody who's in science and still not sure where that path is going to take you, it's okay to not know. It can be very difficult as somebody who is, scientifically inclined, or a scientist in any way, that we like things to be structured, but just lean into the things that you like and seize opportunities, talk to people, build your network, and what is right for you will find you if you put in the effort to seek out the things that make you happy. So as simple as wanting to be around others, seek that out and see where are the spaces that make you feel fulfilled and happy, and those opportunities will come your way. So yeah, it's okay not to know, and engage in science policy. We'd love to see more scientists in the policy space. Absolutely.
Awesome. So again, on behalf of the GSA and everyone in the audience, Marah, thank you so very much for your time today and for your insights and your wisdom. We really appreciate not just what you did today on the stream and in the GSA as an ECLP member, but what you do on the Hill for in advocating for science. Uh,
Thank you.